Yes, we can hear you. We can hear you.
Thank you. So, first, let me tender my apologies for not making Saturday's class last week. So, I actually knew what to teach, but I didn't have any slide as at then. So I had to just postpone this to today. So but today, finally, so I have a slide and then I should be able to like finish up. Probably I should be able to start today and then finish up tomorrow. So I mean just to remind you where we are in our syllabus. So we are now in module 3. So this is where we are. I'm not sure whether you can see this. So this is where we are currently. And I'll be the one taking this. I think within 2 or 3 classes we should be done. Hopefully, my plan is to actually finish tomorrow. But today I just want to like teach you the theory, and then tomorrow we are going to do the practical of today's class. But and our class today is going to be on biological databases or data banks. So before I actually start defining what biological databases are, so I just want to quickly set up the stage by reminding you what the type of data that biologists generate and the kind of data they work with. So we have quite a lot of data that biologists actually generate and work with. The first is the nucleotide sequences, which you guys are, I think, that I should be familiar with. So this is how. So this is like an example of a nucleotide sequences. We also have protein sequences. So this is an example of protein sequences. So this is an example. We also have like 3D protein structures. So there are some times when biologists are interested in the three-dimensional shape of a molecule, most especially probably DNA or proteins. So there are times where they are actually very interested in those. The image. The three-dimensional structure of the protein, and there are lots of techniques that they can actually use to get that kind of data. So I'm very sure that a pharmacy student here should be able to like know an example. So if I should just let me just ask you, can somebody tell me one way you can actually get a 3D structure of a protein or of any molecule? I've taught you in zero, but most of you should actually know. I mean, the equipment that you can use to actually get the 3D structure. Can somebody just tell me one? And this is how your 3D structure looks like. So it's going to look like this. So this is a protein. Any idea? If I want to, let's say probably I isolate a protein and I want to determine the 3D structure of that protein. So what techniques will I use to get that information? Yeah, you can use AlphaFold, but in this instance, I'm just talking about the experimental approach. But AlphaFold is the computational approach of doing that. But I'm talking about the experimental approach. Pharmacy students, do you have any pharmacy students here? I think pharmacy students or biochemistry students should be able to tell me the answer. Nobody. Okay, so I'll just go ahead. Yeah, X-ray crystallography. So you are right, Ramat. So crystallography is one way you can actually. Okay, Adele, you can also go ahead. So we have one more way you can actually use to get that information. So Ramat gave us one, so which is X-ray crystallography, and that was what this Maurice Wilkin and I've forgotten the other scientists used. Oluwa, go ahead.
Oh, okay. Yeah, I wanted to say maybe NMR,
Yes.
nuclear magnetic resonance. I'm not.
Yeah, exactly. So you are correct. So NMR will also give you the 3D structure. I mean, NMR spectroscopy. I mean, those are the experimental ways that you can actually use to get the 3D information. But nowadays, a lot of people are using AlphaFold because AlphaFold most of the time is always very correct. So most people don't actually go through the pain of probably doing these experimental approaches of using NMR or things like that. But I don't think you can use AlphaFold for probably anything that is not protein sequences. I'm not sure. Or I mean, there are still some people that still use NMR and X-ray crystallography, especially if you are working with probably some aromatic compounds and probably carbohydrates or anything that has to do with fatty acid and things like that. But yeah, Adiola, you are correct. So that's the computational approach of getting the 3D structure of the protein. Ramat, you are correct, and Adiola, you are also correct. So those are the 3 approaches that you can use to get 3D information. So the other type of data that we generate also includes gene expression data. I mean, so part of that is, I'm not sure that you can still remember the example data set that we used when I was teaching you unit data analysis. So that's an example of like a gene expression data. So, and also the genetic variation data. So there are times when you are interested in probably studying the single nucleotide polymorphisms. I mean, these are the kind of like information that biologists generate. And there are a lot of ways that you can use to generate them. So there are ways that you can use to generate nucleotide sequences. You can probably do Sanger sequencing or use shotgun sequencing or do like whole genome sequencing. So you can generate this kind of like data. If it is protein, so I've forgotten the techniques that people use to actually sequence, because you can actually sequence a protein as well. But I think one degradation, I've forgotten the name, but you can also do protein sequencing. So if you do protein sequencing, you can also determine the protein sequence, the amino acid sequences of a particular protein. And you guys already gave me the approach that you can use to generate this. If you want to generate a complete genome, you can use genome sequencing. If you want to do gene expression, you can do RNA-seq experiments. You can do real-time sequencing. So they call it, I think, real-time. RNA-seq, I've forgotten. So you can do that one. So you can also use microarrays to actually do gene expressions. You can use microarrays. And if it is genetic variation you are interested in, you can do microarray. You can also use, you can do whole genome sequencing to actually get this kind of information. So that is basically the type of data that biologists actually generate. So, and if you have been with us since we started this class, so one thing we've always been talking about is that the data sets that we generate, that biologists generate, they are usually large. Their file size are usually very large. And most of the time, we don't usually have the computational power to actually store some of these informations. So, because, when sequencing started, and then people started sequencing a lot, then the file size generated by the file size from this biological data. So they were very large. And then so that was just to actually start. Okay, I think I'm already going ahead of myself. Okay, sorry. So these are the data that we generate by biology sorter. And if I actually want to classify this data, so you can classify them as a sequence data. So, for example, protein sequences will be categorized as a sequence data. Nucleic acid will also be categorized as a sequence data, and complete genome sequence will be categorized as a sequence data. And also, we can have some sequence-derived data, for example, gene expression data, and also genetic variation data. These are the data that you actually get from sequence data. So you have to do sequencing before you can probably get this kind of data. So that's why I call it sequence derived data. And we also have structural data. So this structural data, for example, you should be able to guess. So it's just going to talk about data that has structures. And in our own case, we are talking about probably amino nucleic acid sequence structure, the protein structure. And as I was saying before, I mean, because the data sets that we generate are usually very large, so there is a need for us to actually find a ways to store this enormous amount of data sets. And then, so that's actually what now brought about biological databases. So you can just imagine biological databases, so just be like, just imagine them as digital libraries or repositories that hold or house the biological data generated by biologists. I mean, all of this data that you are seeing, all of this data. So you can actually store them in the repository libraries that hold. I mean, so it is usually remote. I mean, you are not, it's not going to be where you are. So you can just store whenever you generate any data and you don't know where to store them. So what most people actually do is they just go on all these databases and then they deposit their genome there. And you can always retrieve it later on. So let's probably are generating a lot of data sets and you don't have your own space. You don't have like a server or you don't have like a memory or like a storage capacity to actually store this information. You can actually probably leverage on publicly available database, deposit your genomes there, and then you can always retrieve them later. But there is always a caveat. By the time you deposit into these databases, the downside is that your data is going to become publicly available, which actually makes sense because majority of the databases were funded by taxpayers. And one thing about the taxpayer is they always wanted to make your data available. So if they are actually stored, so you can store there for free of charge. Nobody is going to charge you. But because nobody is charging you, the only downside to it is that your data is going to become publicly available. So and other people can download the data, reduce them, propose them, do a lot of analysis on them. So that's actually that is basically what I'm trying to say here. So just think about biological databases as like a digital library that holds your biological data and all of this data set that we've talked about. And you might be wondering, so why do we really need databases? There are a lot of reasons that we actually need database. And one of them is data storage, which I already alluded to. I said, there are some times when you need to store your information, your data, and you don't have the ability, so you don't have the space, the memory, capacity to actually, the storage capacity to store these data sets. So you can leverage on publicly available databases, deposit your genomes or any data that you've generated, deposit them there, and you can always retrieve them data. And the second reason is data and information retrieval. So even though if let's say, so I'm not sure probably you guys can still remember what Prof said during our inaugural lecture. When she was trying to advise you guys to learn bioinformatics or any computational skills, as an undergraduate or as a student, if actually you can actually publish, you can just probably go to a database, fetch some genomes or some sequences, analyze them, and then publish your results without even buying any lab reagents or any consumables. In fact, what you just need is your computer and also an access to internet. So if you have internet connection and you also have a computer, you can actually do any analysis without stepping into the lab. And it's not going to cost you anything, except probably if you don't have access to free internet, probably it's just going to cost you your data. So Prof was trying to challenge you that the best way for undergraduate students to see probably showcase research output is to actually probably exploit these databases. You can just go there, fetch any genome or any sequences, and come up with something novel or something new. So that's actually what this one is saying. So probably if you are generating data, like I said, you can store your data there. You can also retrieve it. Unless probably you are not generating any data, you can also fetch. The data sets that other people have actually dumped and uploaded on these databases. You can fetch them and then come up with something novel out of all of these things. So in fact, there are a lot of people that do it, and this is something we actually do a lot when we apply for competition as well. There are two competitions that we've applied for that we actually exploited publicly available data sets that we fetch from public from databases. And it's not about we didn't generate this data set ourselves. We just went there. Some people have generated it. They've generated the data sets. They've uploaded it on a publicly available databases. We just went there, downloaded the sequence data, and then we started analyzing them. So, like I said, databases, we need them for data and information retrieval. So we also need databases to facilitate data analysis and annotation. In fact, so we are still going to show you how this is possible. It turns out that majority of these databases, they actually have inbuilt tools that you don't like. Instead of, you don't even need to probably work on the command line. All you just have to do is probably go on their website and then run some analysis. You can assemble your genome on the public available databases. One example is Pathogen Watch. By the time we start, we will show you Pathogen Watch. So Pathogen Watch is a database, and also you can also use it to analyze your data. You can assemble your genome. You can annotate your genome using the Pathogen Watch, and NCBI as well. So by the time we start NCBI very well, so you can actually see that you can do some blasts, some alignment, some multiple sequence, whether it is local alignment or multiple sequence alignment. You can actually do, there are tools on these databases that will allow you to do all of those analyses. Sometimes you actually don't need to probably even learn the command line before you can actually do some analysis and then publish and come up with something novel. So it is actually what some people actually do. So majority of these databases, they have inbuilt tools that can actually facilitate this kind of analysis and annotation. And also for data sharing. So because like I said the other time, so the reason why databases was also created is to actually facilitate the ability of scientists to communicate among one another and to share their data. So it is possible that you're actually generating the data, and then you can just probably, by the time you deposit it, so I can go there. So in a way, I can go there and then fetch the data set that you've uploaded on these databases. So I can go there, fetch them, and then do some analysis in a way. So that's kind of facilitating data sharing. So can you share data to communicate and to even collaborate. And there are a lot of people that have actually probably strike a collaboration just by depositing their genome or their data sets on publicly available databases. Because sometimes by the time you actually probably upload a data set, and then a researcher somewhere, let's say probably in the US, just see your data set, and then he might actually probably spot something interesting about the data set you've uploaded. And out of curiosity, he might actually just approach you and say, can we take this further? And then from there, you can begin collaboration. So we have some people that have actually, collaborated through sharing information on the publicly available databases. So if you actually want to probably improve your feasibility or probably connect with scientists that are not in your country or where you are physically, so depositing your data set on databases can actually facilitate that. So, and you can actually probably be the beginning of something great for your career and things like that. So there are a lot of people that have actually benefited from that. And for knowledge preservation for future use, sometimes, people just even deposit all of this information on database without even knowing the immediate importance of some of these things. But it will probably be the future generation that will just come and say, wow, I think we can actually come up with some of these things. Just imagine this AlphaFold. Before, the AlphaFold kernel was not even, before it was actually invented. Please, can you still hear me? Or have I lost communication?
Yes, we can.
Yeah. Okay. So regarding the AlphaFold, if you actually read about the AlphaFold, so these data sets that were used to train the model were gotten from sequences that people have started depositing in the 1980s, 1990s, when all these sequences, all these protein sequencing and Sanger sequencing or shotgun sequencing started. So, let's probably, at that point when, around those 1980, 1990 something, I'm very sure that they probably didn't know that probably there will be an AI revolution that will just come, and then we can now start predicting protein structure just by the sequencing. They had no idea about what they're actually doing. They were just depositing there, probably for data storage, data retrieval, to facilitate data analysis, data sharing. And then, so it was from these historical data sets, the data sets that people have been depositing since years back. So that is what, the AlphaFold model used to train their model and then come up with something novel. So that's what I'm just trying to like say. Sometimes you just even deposit without even knowing why you are even depositing it. But later on, somebody can just come up and just, okay, can we even think about, and then come up like a very novel way of thinking or approaching a problem. And this is actually an example of knowledge preservation for future use. So then people that were depositing protein sequences, they probably had no idea that their sequences will be used to train a model that can predict the structure of the protein. They probably had no idea. But look at where we are now. AlphaFold model cannot even predict very well. And most of the time it's always very correct. So that means you don't even have to like, because all this NMR that you are seeing and all of these crystallography, they are very expensive techniques and they are very laborious. But now, without even if you don't even have any resources or you don't even have any funding, once you shall have your protein sequences, you can actually predict the structure of that protein and then even, talk more about your analysis. So that's actually, the reason why we need databases. Just for data storage, data, and all of this information are itemized. Then, so this is where we need to be very careful, because I won't lie to you. When I was actually reading up, I was confused because I was seeing lots of different classifications. But of all the classification I saw, this is still the only thing that makes sense to me. So I just had to like come up with this. I had to like draw all of this thing myself, just to be able to like capture everything that I have read from all this online stuff and by torturing ChatGPT as well. So we have to be very careful here, because although this is a classification that makes sense to me, it is possible that if you actually go online, you will actually see a complete different classification. So if that is the case, don't be surprised. In fact, like I said, this area is kind of like, it can be confusing sometimes, but I will try my best to actually explain. So all these databases that we are seeing, we have many of them. Like I said, some of them, when I was telling you about the kind of like data that we generate, if I should go back, I said these are the kind of like data that we generate, right? It turns out that, there are databases that just house, what they just do is just to house any nucleotide sequences. So that's what they do. I just want to be storing nucleotide sequences. And we also have another, like a separate database to just store and analyze protein sequences. And we have another different database that's the sole function is just to like store three-dimensional protein structures. And we also have some other databases that are dedicated for complete genomes, others for gene expression, and another for genetic formation. So by the time we actually delve deeper into today's class, we actually see that we have a lot of databases, a lot. In fact, I would say we have at least like 30 different databases. Some of them are actually, for nucleotide sequencing, you might actually be surprised that you might actually have like just probably 15 different databases that are storing the same thing. So we have a lot of databases out there. But out of all of these databases, we have some that are very, very popular. And that is because they are well funded and then they are well created. We have dedicated scientists, dedicated researchers that are actually maintaining these databases. And there are some databases that somebody just created them and then no follow up. So no maintenance after that. So we have a lot of databases. So you have to be very careful out there which databases that you use. So most scientists, so they tend to like use databases that are well created, up to date, and maintained. So So you actually see that despite the fact that we have a lot of databases, some databases are much more popular than the others. I mean, among scientists or biologists. So we can classify databases. Like I said, databases are just like a digital library that houses all of these data sets that I talked about. And you can classify them based on nature. Let's say you want to probably classify your databases based on nature. I mean, the nature of biological information, whether is it a sequence database or is it a structure database? I mean, is it a database that is dedicated for storing sequences or is it a database dedicated for, I mean, structure or functional information or expression data or operation data? I mean, we have a lot of databases. So for the sequence database, we actually see that for the sequence database, you actually come here. I'm not sure whether you can see my cursor. The sequence, you can see if it is protein sequences, you can see these are the databases for protein sequences. If it is nucleotide sequence, so this is, I mean, the one that will store nucleotide sequence, anything that has to do with nucleotide, gene bank, DDBJ, short read archive. I mean, they will always store DNA, anything that has to do with DNA, and if it is protein, anything that has to do with all these short chain amino acid. So these are the this is a popular database that will actually store down. So like I said, we might actually have others, but what I just could say they are the most popular ones. So they are very popular. So, and then if it is structure that you're actually interested in also, you can store them in protein databases or CSD. So we have a lot of databases, like we have ProDOM, ProCite, PFA, a lot. So, and if you're not careful, you might actually be overwhelmed. But in our own case, we just focus exclusively on probably the NCBI stuff. But I will soon get to that actually. So you can classify your database based on the nature of biological information that you are storing. Is it a sequence data set that you want to store? Or is it a structural data that you want to store, like a three-dimensional structure that you want to store? Or is it like a functional data set that you want to store? If you are interested in that, probably cake is very popular. You might actually have eggnog. So we have lots of databases. If it is expression databases, you want to probably put it in Gene Omnibox. If it is variation, you want to probably put that one, probably database for SNPs. So that's actually the SNP database. So that's actually what this one is talking about. And you can also classify your database based on the content and what type of data are they storing. Is it experimental data? Experimental, by experimental, I mean raw data, the one that you actually generate in the lab. So for example, probably the one you generate from whole genome sequencing or shotgun sequencing, or I mean Sanger sequencing. So or Fredman degradation or anything. So if that's the case, so we'll classify them as primary databases. So primary databases only holds, so they hold data sets that are generated in the lab, that are raw. You've not analyzed them. They are just raw data. I mean, data that's not been analyzed at all. That is, and you've not created them. I mean, organize them into information and things like that. So that is what this primary databases holds. So, and if you actually go to this flow, I mean, this diagram, what you can actually see is if I actually come here, so biological databases. So I first categorize them based on what the contents. You can see primary databases, secondary databases, composite databases. So by secondary databases, so secondary databases are the databases that you, so these ones, they are derived from primary databases. So all of this raw, I'm not sure, like, if you can still remember the definition of primary. So they are raw data. I mean, the old data sets that are raw, that you've not analyzed. So the analyzed one, so the data set that you now took from this and you now analyze them, you analyze them. So that is what we classify them as a secondary database. So you have to analyze. So in a way, so they are no longer raw. So they are created, analyzed data sets. So any data sets, any primary data sets that has been analyzed and curated. So they will categorize them as what secondary databases. So derive, analyze, and annotate data based on primary data. So you always probably generate this information from. You get secondary databases from primary databases. So you need the information, the data sets in primary databases before you can probably come up with like a technology database. And for composite databases, so just imagine a composite databases as, I was telling you that we have all of these isolated, different databases. One example is Uniprot, Tremble. Another example is GenBank, AMBL, GDBJ. Another one is PDB, CSV. Another one is Prodom, ProSite. All of these individual databases. So composite is just, so because they are isolated, so they are not really speaking, talking or communicating with one another. So what composite database is just doing is just going to like bring all of these isolated databases into one, under the same roof. One example is, let me just show you one example. One example is going to be this. If it actually goes to NCBI, so NCBI is a popular composite. So NCBI is a popular example. So NCBI is just, so if you actually come to all resources and you come here to databases, you can see a lot of databases here. So this is another, we have assembly databases, we have bulk collection databases, we have bulk project databases, we have bulk sample. So they are just isolated. What NCBI just came, when NCBI was actually, set up, when it was established, the main task their main objective was to actually find a way to bring all of this information together. So just see them as like a database that is trying to like harmonize all of these individual databases. So that is actually what composite databases trying to like do. So another one is Embu EBI. So that's another example of the composite databases. So inside these databases, you will always have access to different databases. So that's actually what composite databases are. So please, do we understand up to this point? Do we understand up to this point, please? All right. So it's only, okay, Victor, please, do you understand? If you don't understand, so this is the best place to probably ask your questions before we actually delve deeper. Do you understand up to slide number 6? Okay. Others, please, do you understand? I'm only saying Victor, so okay, so they under, please. So, okay. Let's just probably talk a little bit about this flow diagram. Like I said, everything. So this is separate biological databases, so I can umbrella term. So under biological databases, we have like 3 different type of databases based on their content, whether they use experimental data sets or not. They hold experimental data sets, so they will be categorized as primary databases. And if they don't, if what they hold is an analyzed or like a curated data, so you will categorize them as say, secondary databases. And composite databases are databases that tend to like consolidate, bring or harmonize all of these different databases together. So you're just going to bring them. So instead of probably clicking 20 different databases, so you can always find the link to all of these 20 inside one link. So just see this as a database of other databases. So that's actually what composite databases are. So, and if it's a primary database, like I said, when we're talking about the kind of like data that we generate, we said sometimes biologists generate protein sequences. They also generate, nucleic acid sequences. Sometimes, so we also generate structural information, but especially protein. We generate complete genomes. So based on this, you can also classify. So if it is or is this sequence or structure? Although you can categorize them into more than this, but the most popular ones that we'll be talking about is just sequences and structure. If it is sequences, it could either be protein sequences, it could either be nucleotide sequences. If it is protein sequences, I mean, if you want to deposit your genome or you want to find information about a protein primary sequences, so we want to go into probably UniProt and UniProt knowledge base Tremble. I mean, this one is translated EMBL. So you want to come to this database for information about protein sequences that were experimentally generated. So you want to come here. So let's probably regenerate any protein sequences in your lab and you want to deposit. You come here and deposit here. And although this is just an example, we have others. The other ones could be, I think, another one is probably protein information resource PIR. I just noted this one because this one is very popular. So, and if it is probably what you do, probably you don't work with protein in your lab. For our own case now in our own lab, we hardly work with protein. So most of the time we're always working with DNA sequences. So whenever we generate DNA sequence and want to deposit, want to deposit into GenBank, or if it is short-read archive, you can put it in short-read archive. So if that is NCBI, we are talking about NCBI. Or let's say probably you are talking about EMBL, or you want to put it in European Nucleotide Archive. So we have a lot of databases. So, and if it is structural information, we know there are some labs, especially the labs in Austria, because of what they do is just structures. There are some labs like that they're always interested in structure of the proteins. I mean, they're always in structural biology. So they call themselves structural biologists. So they're just interested in structures. So probably generate or they isolate any proteins and they crystallize them, then subject them to probably the proteins to crystallography or NMR spectroscopy or any imaging stuff. You want to deposit that into probably PDB, so protein database or CST. Because these are the data sets that you generated experimentally. They are fresh from oven. You've not analyzed them. They are raw. They've not been created. You just deposit them here. So by the time we now graduate to these secondary databases, so secondary database, what they are just doing is, okay, probably, because we said all of this information, they are raw, but before we can start making meaningful information, as a scientist, we always want to derive insights, we want to turn data into knowledge. So that's actually what leads to secondary databases. So they've been created and be analyzed. So I mean, some people just came and said, okay, why all of these data sets, they are just raw. They don't have any meaning. So how can we make them meaningful? So, and then started and they came, okay. So there are some people, like I said, I told you that there are some people that are just interested in sequences, whether protein or nucleotide sequences. Then these people that are interested in protein will go to UniProt Tremble, take all of these raw protein sequences. They take it and then they do all sorts of analysis, probably multiple sequence alignment or things like that. So they do all these multiple sequence alignment just to see, the regions, the motif that are conserved, or probably you're interested in studying the motif or finding the motif within the protein. You have to first align them using multiple sequence alignments. Or else probably wants to even know the domain of a protein. I'm not sure whether you guys know the difference between a motif and a domain. So please, if you don't know, go and find out. So there's a difference between the motif and the domain. So, and there are some people that's what they are just interested in. So they want to know, okay, what is the motif in this particular protein family? So what is the domain within this? All of these protein sequences. What could be the domain? So they will come here and then they turn it into something meaningful. And so wait, this part is conserved, this part is not conserved. So things like that. Then, if probably you are interested in nucleotides, there are a lot of databases here. I just itemize all of these things. I just put this because they are kind of popular. SciGuard, all of these things, but we have a lot of databases that fall under this nucleotide. So what they just do is, okay, they'll just take information from GenBank or EMBU or DDBJ. They will just take information from them and then analyze them. So this FlyBase is just like a database that contains information about anything that has to do with flies. Just imagine any flies. I mean, things in probably kingdom, insecta. Is it kingdom? The class insecta? I don't even know. Yeah, I can't remember my zoology. So yeah, all of those things there. So they just so that's what this fly-based database is just talking about. All the information regarding flies, whether Drosophila, anything, any flies, housefly, dragonfly, anything fly that you can think of. So you will find all of those information here. So I mean, those are just people that are passionate about flies. There could be some people that are probably passionate about probably rodents or mammals. So they have their own database also for mammals. If you just go there, so all of these databases and all of these data sets that people have generated, deposited regarding mammals into GenBank and Kine, they took everything, they analyzed everything, and then come up with something meaningful. I mean, they turn it into knowledge. All of these data, they turn it into knowledge. So you'll find all of this. So that's actually what's happening here. So they are just taking information. So the protein, the secondary database, secondary protein database, we take information from here, turn it into something meaningful. The secondary nucleotide database, we take information from here, turn it into something meaningful. And if it is protein, the secondary structural databases, we take information from here and then turn it into something meaningful. Do we understand it? And then, like I said, composites just contain all of these things, all of these different, whether they are primary or secondary, they just put all of them. So instead of probably clicking different link, like I said, there's a different link for this, there's a different link for this. Instead of clicking all of these different things like that, you just probably bring everything together inside the same house. So that is what NCBI and API are doing. So they are just repository containing other repositories. So for these two that I say. Any question up to this point? Any question? Any questions? So you guys, you understand everything now. Also, I'm not asking any question. I actually expected you guys to be confused. I'm surprised that you're not asking questions. Okay. I know there are some people that are just on this call, but they are not with us. So we are just here to mark attendance. Okay. I see some people, they've not even responded to me. Okay, let's continue. Like I said, I have just 11 slides, so we should be done very soon. So, like I said, if you come to this diagram, you know, when I started, I said we have a lot of databases. We have a lot, at least more than 50. We have a lot of databases. So what I just did was also itemize the most popular. They are popular because scientists have adopted them, number one, and because they are well funded. So most of these databases that you are seeing here, most of them are funded by the government, most by probably European governments and US governments, and probably Japanese governments. So majority of them, so they are, I mean, they've been well, I mean, they are well funded, all of these databases, and then they are always curated. I mean, we have people that are dedicated, we have people that have been employed to actually maintain them. So there are people that are always there monitoring things, trying to say, okay, is there an error or not, things like that. Because sometimes people actually deposit rubbish. So there are some people that are there to actually vet out things that are bad. No, Nigeria governments. So actually, Nigeria governments, they also have should I say we have it? We don't. I don't think we have a database. We don't have a data. I don't think we have. I'm not sure. Sure. So it's possible that we have, or probably I don't have the information. Oh yeah. So for these, I just itemize. I mean, the ones that are very popular. But we have others. In fact, we have databases that just holds anything that has to do with literatures or articles. I'm very sure that you guys are already probably familiar with it. So one example is PubMed and one example is Medline. So these are very popular. We have others, but these are the most popular, Medlines and PubMed. So this is a database and this is, so they just hold these articles or that has to do with probably biology or things like that. They might also hold information regarding some other things, but contain articles and research paper on different journals. So if you come to this bibliographic databases, example is Medline, PubMed. So they're an example of bibliographic. And you might be thinking, well, are this one really protein sequences or sometimes you need to like probably read literature before you can even probably analyze your data set sometimes. So I mean, and one example of a databases that scientists or biologists actually use is Medline and PubMed, because although they might not actually contain sequences or things like that, but sometimes they contain links to those sequences, and they even contain information and experimental approach. I mean, like the methodology that the researcher or the author used. So that way you can actually probably vet something that is actually rubbish or garbage and something that's actually authentic and accurate. Just by reading the methodology and the approach that these scientists that deposited probably any of these sequences or 3D structures or all these data sets that we've talked about, by just reading their methodology, you can see, okay, whether this is rubbish or not. Like I said, majority of these databases, the old nonsense, I mean, garbages sometimes. So if you are not very careful, you might be thinking probably you are making sense of your data. I mean, you are, I mean, synthesizing or creating knowledge from this data set. But whereas what you are doing is you are just analyzing garbage. And there's one concept in bioinformatics, garbage in, garbage out. So if we are analyzing garbage, what we will get at the end of the day is also going to be garbage. So this is, so although we didn't talk about this, but please be familiar with this. We've talked about OKNCBI, so you know about this. DDBJ, we've talked about this. The Clota database, we've seen this. YouTube, like I said, KEGG. So this one is going to hold anything that has to do with your probably, I mean, your ontology or your metabolic pathway stuff. So this way you can find information regarding. Let's probably you have a gene and you want to know, okay, this gene, which biochemical pathway is it? I mean, is it implicated? So just come to this database, and then you can be able to like see, oh, this top. I'm trying to like remember one of the first enzyme in all this our is it Krebs type or glycolytic pathway. I can't remember. So all those fructose ones, commerces, diphosphates, I can't remember. So if you want to know any gene or any protein that is implicated in metabolic pathway, just come here. So, and if it is enzyme databases, I mean, if probably, I mean, you want to get any information regarding an enzyme or you want to deposit an enzyme, or probably you discover a new enzyme and just probably deposit it in enzyme database, and probably you want to know more about a particular enzyme, you can also come to this database, which is XPassy Libase. So these are, I mean, the two popular databases that holds information regarding anything that has to do with enzymes. And we have disease, Mendelian disease. So this one is strictly for, I think, human anything that has to do with human disease. I think you will find it a disease-related information. So people that have probably congenital diseases, or probably you have like a mutation and things like that, and then you want to look, okay, this mutation, if this mutation should occur in this particular gene, so what kind of disease can that person actually come up with? You will find that information to be. I think online Mendelian, it means online Mendelian information. I can't remember. So OMI. So if it is probably, I mean, chemical that you are interested in, like I think probably people that are interested in docking and all these lots of chemicals, I mean, Fanchem and other people in chemistry. So they like to play a lot with this database, PubChem. So think of any structures that you can probably, I mean, that you can think about. So if you want to still kill, probably you even discover a new structure. You want to see, has somebody discovered this structure before I declare victory that I've discovered one structure that nobody has discovered before. So you first want to, I mean, you want to come to this chemical database and then probably deposit it there and see whether you will see any match. So if you see any match, so that means, I mean, what you've discovered has been discovered before. So it's not really your own discovery. And if you don't see any match, so that means probably you will be you can take ownership or claim ownership of the structure. So PubChem, I mean, people in docking, molecular docking, so they like to play with this particular database, PubChem, and you will find it on NCBI. So, and if it is gene expression data that you're interested in, or I mean, you'll find it in gene expression omnibus, human gene expression. I mean, there are a lot of other databases. Like I said, although I say itemize all of this, we still have others. So this is not, I mean, these are not exhaustive. So we have a lot of databases. We have a lot, and it's your job as a biologist to know which ones are actually good, which ones are not. You need to be able to. You need that filtering mechanism to be able to like tell the bad databases from the real one. Yeah. So the composite, I just wanted to talk about the composite databases more. Like I said, the unified different isolated databases, all of those databases that we've talked about. So what composite databases does is say those isolated, bring everything together under the same roof so that we don't have to probably be jumping from one link to another or things like that. So if you just probably log in to NCBI, so you can get the link to other databases just by being in NCBI. Somebody asking a question. So, like I said, DDBJ. So DDBJ means DNA Data Bank of Japan. So that's the meaning of DDBJ. I said it's not really a computer database because so what you can always deposit is just DNA sequences. So it's just DNA sequences. Okay, we have questions. Okay, so let me just respond to Akitayo's question first. I have the question, it's asking a question, is it that I can access all the primary database and secondary databases there? I don't really get that complete database too. Yeah, so that's exactly what I'm talking about. So you can access, so like I said, I'm not sure whether you joined I think probably you joined the class late. So this is NCBI. NCBI is an example of a composite database. And if you come to this resource, I mean, if you click on all of these resources and then come to these databases. So this is one database. So this is a link to one database. So genome database. So this is a link, and this is another link to another database. So these are just different databases that has been brought together under the same umbrella. So that's actually what composite is doing. Although there are some, I mean, you might not probably get information about probably UniProt, and that is because there is politics at play here. So before I even move on, so this thing that you are saying, DDBJ, like I said, this databases was created by the government. This project was funded by the Japanese government, and this one was funded by the European government, the European Union, and this one was funded by US. You can see, I'm not sure whether you understand geopolitics. So everybody is trying to like take ownership of their own data. In fact, African, like I can even tell you that African CDC also, they are trying to like come up with their own database so that it's just going to contain, I mean, any data from any member of the African Union states. So by the time it is fully developed. So everybody is trying to like take ownership of their own data. So the government in Japan said, okay, I mean, I want my citizens to be depositing their genome in DDBJ. I mean, this is the name of the database. So this one is for the European Union, Europe's, and this one is North America and the US. Although this seems to be very popular now. In fact, there are some European countries that they don't even deposit here at all. They don't deposit here. In fact, I have one mentor that she's always saying why is it that most African countries don't like using embu. This is this is very popular among biologists worldwide. But so I think followed by this, then followed by this. So NCBI is very popular. So there are some databases. So this embu is a composite database. So it's going to contain probably databases. Let me just show you embu. I think it's EBI. I think it's EBI. So this is embu. So this is from the US government. This is from the European Union government. So these are if you come here, look at all of the different DNA, RNA. So they contain the same information. I mean, you can see sites. So this one has like a different site, different links. The links to this, so all of these individual databases are different from this. So if you actually click on probably RNA or DNA, something like that, so you come here, GWAS. So these are the databases that falls under this category. So GWAS, International Genome Sample Resource, ANNOTAB, if you want to do BLAST. Like I said, databases, you cannot just store information on it. You can also use them to do analysis. So one particular tool I can use to do analysis is this. So you can see this one is a data resource, this one is a the green categorizes. So this one is a 2. So it's not a database, but a 2. So this one is a 2. So this one is also a 2. Cluster omega. You want to do multi-sequence RNA. This one is a data resources. So they provide description. So although they might actually be holding the same information, but there are different databases. So it's possible to have like different databases that do the same thing. They are doing the same thing, but they are different. So it's just like Apple and Android, and let's probably Redmi. So Apple is creating phone. Redmi they are also creating phone. But at the same time, they're all doing the same thing. Mobile phone. So that's what they are just doing. So they're all creating mobile phone. Apple products. We also have Redmi products, right? But Redmi belongs to Chinese government. Apple belongs to, the US. The owner of the company is a US, is an American. But they are doing similar thing. So that's exactly what is going on here. So NCBI belongs to the US government. embu belongs to the European government. So they are capturing the same information, like anything that has to do with all these DNA sequences, all these things that we've talked about. All these things that we've talked about here. So they are all capturing probably all these data sets. So they are all capturing the same data sets, but different databases are responsible. So probably with this protein sequences here, for example, let me just show for nucleic acid, the databases in NCBI that is actually doing that is GenBank. And for European government, the equivalent of GenBank for European government is embu. So it's embu. It's still the same thing, and the equivalent of that for Japanese government is DDBJ. I'm not sure whether you understand. So they are all capturing nucleic acid sequences, but if it is US government, so they are capturing it in GenBank database. For European governments, so they are capturing it in embu database, and for DDBJ, they are capturing it in DDBJ database. I'm not sure whether you understand the explanation, but they are all capturing the same thing, just in different flavor. Okay. So Olariwaju just said, are there ways or metrics to identify database whose information are not accurate? Or do we just make use of most popular? Yeah, sometimes something is very popular among scientists. So most of the time, so I'm not saying probably that is the best. So most of the time, it means is you can actually use it as a good signal or proxy of probably uh, accurate databases. Actually, if something is actually very popular among scientists, most of the time it means, I mean, it is well created and well maintained. So most of the time, you can just use that one as a metric, like a proxy. But I don't think I know a better way to actually vet out, I mean, bad databases. But most of the time, so bad databases that are not usually maintained. So they're not usually maintained. So, and yeah, sometimes popularity among the scientific community means, I mean, good, good. But yeah, I'm not sure whether I answer your question. Then Joel said, please, is it possible that two databases give different information about the same gene? And how will we know which one to trust? Yeah, it is possible. It is possible. What you need, so it's just like CLSI standard and UCAST. So all you just have to do is probably reference, and then most of the time, like if you actually probably read very well, most of the time, you actually notice that the two database may actually they may have like good and strong evidence for their claim. But most of the time, so we'll soon get to there. Most of the time, we don't usually have like discrepancies, and I will actually tell you why in just a little bit, Joy. So I will answer your question when we get to that slide. So yeah, I think this slide is going to answer your question. Like I said, NCBI is owned by the US government. EMBU MBI is owned by the European government, and DDBJ is owned by the Japanese government. These are the what databases that you can find under NCBI. We have the GenBank, BioSample, BioProject, BLAST, PubMed, SRA, WebSeq, taxonomy. So you will find anything. If it is probably you are interested in classification, let's probably you have an organization, you want to know the classification, just go to the taxonomy database. So these are database. And you can see dots, dots, dots. So this dot, dot is saying we still have others, but because I mean we don't have a lot of space, so we just so that I mean this dots, dot, dot just means and so on and so forth. etc. So under EMBU, so we have ENA, we have biomodels, we have expression atlas, we have enzyme, and then one Uniport, we have one base. You can see there are different databases, but they might be doing the same thing, especially GenBank, ENA, and the DBJ. So this particular, the first row, they're actually doing the same thing. So because they noticed that, I mean, this, because, I mean, NCBI, then NCBI was just doing its own. It was not communicating with EMBU. It was not communicating with the DBJ because geopolitics was at play. Everybody was trying to like take ownership of their own data. But the scientific community noticed that, I mean, this is not actually helpful to the broader, I mean, scientific community. So they came together and then come up with this initiative. So, initiative. So this initiative is called the International Nucleotide Sequence Database Collaboration. So we just brought these different, I mean, composite databases together and say, I mean, can we be communicating together? So the knowledge or the data that you generate, can you make it available to this and to this? So that I mean things are not just probably so that I mean knowledge is not just probably isolated to a particular region. So what's that? So this collaboration, what's the output? I mean the outcome of this, I mean collaboration is that we now have information that's now being shared regularly between, I mean, these different databases that you are seeing. So if you deposit, like, so that means if I, as a scientist now, if I should deposit in NCBI, and then I didn't deposit here, I didn't deposit here. So let's probably I just choose NCBI. The information I deposit is automatically automatically going to sync with this thing. So NCBI will transfer it to Embu and also transfer it to the DBJ. So if we just, so whatever information that you dump here, automatically is going to like sync. You will find it here, you find it here. So that means anybody that is choosing probably DBJ. So probably the Japanese people or the people in Asia, if it is the DBJ that they are interested in and they deposit it here, you will find that information later on in Embu and also in NCBI. So they now communicate together now so that, I mean, whatever information that I generate here is automatically going to what? To show in the other databases. Do you understand this initiative? And Joy, does that answer your question a little bit? So that way, so that we don't have discrepancies, but there are still some times when discrepancy might actually come because, I mean, it's not, there's some databases that are here that are not here, and there's some databases that are here that are not here. So sometimes it's possible that, I mean, the information that is here might not, but you shall get the gist. Most of the time, they're always communicating with one another, these three databases. So they are no longer isolated. So such that whatever information or data that you deposit here, you automatically find its way on the remaining databases. Do you understand up to this point? All right. So I'll just continue. So personally, although like this is just my own personal stuff, there are some people that they are comfortable working with Embu API. There are some people that are comfortable with that. But personally, I like using NCBI. So I'm not saying probably you should also learn NCBI. I mean, as I will even probably put it to you that you should actually explore these three databases, especially DBJ, Embu API, NCBI, and then choose the one that you are comfortable with. But for me, I'm comfortable with NCBI. And also, this is what I'm so sorry. So this is what I will be teaching you as well in subsequent classes. So because I'm comfortable with NCBI. So it's possible that probably if you go to the UK, probably you get a scholarship and then you go to the UK or you are being tutored by a UK scientist. They might not actually know how to use NCBI. So it's possible that what they will be teaching you is Embu, how to navigate Embu, how to use the tool, how to deposit, how to fetch data, how to do all sorts of things. So it's possible. But for me, I was taught using NCBI. And I will also be teaching you how to use NCBI. But you are also free to explore the other flavors of databases and then choose the one that you are more comfortable with. So, I mean, this is the signature, I mean, homepage for NCBI. So, and this is their logo. So you can see the National Center for Biotechnology Information. So it's just like a division under the National Institute of Health in the US. It's a division. So, and I will just demo just a little bit. I'm just to interact with it. I'm not going to we are not going to do the practical. So tomorrow's class is just going to be for practical. I mean, how you can do some. There are a lot of things that you can do on NCBI. There are a lot of things. But I'll just show you some little functionalities and then probably point you to tutorials that you can actually watch if you want to delve deeper. I'll just show you the ones that we think will help you in subsequent classes so that by the time we start, if it's okay, go and do blast, go and do this thing, you should be able to find your way. So, but I won't explore the full functionality of NCBI, but believe me, NCBI is very, very powerful. So there are some people that they don't even know how to work on the command line, that everything that they do is just NCBI, and they publish a lot of high quality papers. They do a lot of high quality stuff just because they know NCBI alone. NCBI is very, very powerful. So this is just the link. All you just have to do, I'll just probably click this, is just type NCBI. If you just type NCBI and then just click on this, you can see. So this is the home page. So you can see that Trump is really messing up with NCBI presently. You can see their message notice because of a lapse in government funding. The information on this program may not be up to date. Transactions submitted via the website may not be processed, and the agency may not be able to respond to inquiries until appropriation are enacted. So I think probably they have this back and forth already with Trump administration. Hopefully they sort it out. But they're just trying to like tell you that, and I told you that this is a well-created database, computer database, is well-created. They employ people. They employ people that are in charge of maintaining this database. So this database is usually very up to date. Is up to date, and is almost. I don't want to say probably it's very accurate, but it's. I can see it's accurate. Majority of the information that we find are usually accurate, especially the one that has been created. So, and then even if they are not correct, so they can always, if probably they find out something new or wrong about their previous stance, so they are going to like correct it. So they employ people because it is maintained. So that means they will need a lot of people to actually be in charge of this maintenance. So there will be some people that are in charge of probably policing each of the databases, the one in charge of probably all of these functionalities. But I'm not sure whether you guys are acquainted with US politics. So some government is trying to like cut down expenses. I mean they don't want to have like a large government. So they are trying to like uh is it they're trying to like sack some people, things like that. So because they want to like reduce the size of their government. And though it's possible that NCBI also are affected. So they're trying to like cut down the number of people that are working for government. And you might actually be thinking, well, is this a good thing or is it a bad thing? There are sometimes when it can actually be good because no government actually wants to have inflation in their. They don't want to have inflation because the more government, the more people that you employ, so the more money you have to spend because you have to pay their salary, cater for their revenue and things like that. So the more money you are spending, and if you are spending a lot of currency, if your currency is flooding the market, so the implication of that is that your currency is going to like lose relevance, and then so that's actually what causes all these inflations and things like that. So I think probably these governments are, the NCBI is affected. And I'm not sure whether you guys are also very familiar with what is going on. So the Congress is not accepting, the budget. So the Congress is not accepting the Trump administration proposed budget for things like that. So a lot of workers has been followed. They've been forced to like stop going to work temporarily. It's not something that's going to be permanent, but a lot of people are not going to work because they are not being paid, and that's because the Congress is delaying, budget allocation, approval, and things like that. So I think probably that is what is happening. So that's why they put these two things. It's possible either of those cases, say that probably Trump administration sacked some people, or because of this temporary, back and forth between Trump administration and the Congress, the US Congress. So, but politics aside, so this is NCBI database. You can see there are a lot of links that you can click. There are a lot of links. So we'll still explore this one. You can see that this search engine that you are seeing is a very powerful search engine. It's a very powerful. We call it the Entree. They spell this Entree search engine. Entree. So it's not Entree. This Entree is like a French word, and they call it on, like just on. So it's pronounced Entree. Entree. So you can see it is very, very powerful. So if we need to like search anything on NCBI, we want to search anything, probably a particular gene or anything. So this is what you should be using. For example, let's say I want to search Homo sapiens, anything that has to do with Homo sapiens. You can see. So it's going to give me information about Homo sapiens. You can see. So if you go to literature, so this is a database, Bookshelf database, mesh database. So you say there are records of that 22, 92,120 records of Homo sapiens in this particular database. And we have 5 in mesh database in NML catalog database, we have 287 instances of Homo sapiens, things like that. And then you can see it's a very, very powerful database. And if it's protein that you're interested in, probably you're interested in Homo sapiens protein. So it's probably interested in the conserved domains of probably proteins from Homo sapiens come here. So anything. So if it is PubChem, I mean the chem is probably the proteins or the compound, any compounds generated, I don't know, by humans. Probably is where you find them. A lot of it's very powerful, this search engine. So we are going to like explore how you can actually search, because this search is different from your Google search. So this one has been optimized for searching anything on the databases that are hosted by NCBI. So I mean, it has been optimized for that. So if you say you want to use Google to search it, so it's Google, it's not going to be efficient for that. Yeah, Victor, please go ahead.
All right, good evening, sir. When you search for Homo sapiens, it said result found in 28 databases, but then I could count just think 6 or 7. So I don't know where the other ones are.
So this is one database, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29. So these are the databases. This one is just like a category. It's saying, okay, so these are the databases, bookshelf, mesh,
Okay.
NLM. So you are not counting this. So this one is just like a broad categorization that, okay, all of these databases, they are just talking about bibliography, the one we talked about here, about literatures. So it's just trying to categorize them so that, I mean, you are not confused. And you say probably you are just interested in the genes from Homo sapiens. So just come to here. So this is where you find information about the genes regarding Homo sapiens. So it's either you come to this place, or you come to this place, or you come to this place. So these are the so these are just broad categorization of these databases. I'm not sure whether that answers it.
Yes, sir.
Yeah. So this is the NCBI homepage. So what I just want to show you is the NCBI homepage. And then you can see on the right hand side, you can see a lot of resources. So it has categorized everything for you. That's categorization that we saw earlier, all of these things. So you can find them here as well. Let's probably you are just interested in anything that has to do with chemicals or bioassay. I'll just probably click here. And then, so this is where I will find anything that has to do with biochemical stuff. You can see anything that has to do. So you can come here, computational. So these are the databases, computational resources from NCBI structure book, PubChem bioassays, PubChem compound, PubChem substances. And let's probably it is DNA and RNA I'm interested in. Just click on this. And then, okay, well, I should know that, okay, well, assembly, I should be looking at assembly, bulk collections, bulk projects, bulk samples, consensus, gene bank, this and that. So, and if it is proteins that you're interested in, I mean, you need probably information about any proteins. So that means bulk projects. Yes, probably want to deposit, you want to download, just come here, conserve, all of this. So these are all of the things that you can. And I mean, let's say this one is saying if you want to download anything that has to do with protein. So these are the tools that you can use. I mean, these are the tools that can work with any sequence that has to do with proteins. You can see we have so blast, virtual interest, cobalt, CN3 conserved domain. We have a lot of things. So my assignment for you is after this class, just click on NCBI and just play around. You don't need to like probably, yes, I mean, just play around and even just get acquainted with, I mean, get yourself familiar with a lot of these things. So that is one way to actually probably get into all of these things. But the other way is probably just to come here. If you come to all resources, also, you can see, you click on all resources. So this way you find the list of everything that we have. To NCBI, the list of everything that we have. And that's why we just want to like click on the databases. That purpose, you are just interested in databases. So this is where you find anything that has to do with databases. So these are all of the databases that are on NCBI. And let's probably want to download. So you can download using this tool. or FTP. Let's probably want to download over the internet. So yeah, and download using all of this. And let's say probably want to submit because, like I said, I told you that if you are generating data sets and you don't have the capacity, the storage capacity for storing, you can actually take advantage of this and submit into. So depending on the data set that you have, so just click on it. Let's probably are interested in that projects. And just click on this. Anything. So you can see Gene Bank. You want to click on Gene Bank. So there are three ways: Pancake, barcode, things like that. So and the tools, these are the tools that you can use on NCBI. So these are the tools you can say Blast, Microbial Genome. So these are the tools Blast or a lot of stuff here. And how to, let's probably you need tutorial. So you want tutorial, okay, how do I do this thing? How do I submit your data? So you can read up. I find information about Glycan, you can read up. Download NCBI software, you can read up. So this is well-curated database, a very well-curated database for that. Yeah, so let me just go back to the home. So remember, this is the home. And let's probably you need tutorial. So if you need tutorial, I think this is where you can find that. I believe you find. So like I said, I told you that you cannot explore what you can do. I mean, for this class, I mean, exploring all of NCBI is beyond the scope of this class. So we'll just show you the one that we think is relevant. I'm not saying other ones are relevant. I mean, relevant to Bioinfo UI. I mean, so just teach you those one. But as a diligent student, they advise you to actually explore. So if you actually want to explore further, you can just probably come here, read the documentation or tutorial. And they also have like books. In fact, if you actually want to read any books, you can come to NCBI. I mean, it's not, I don't think it's here. But I think they have a bookshelf stuff that you can actually read. But for tutorial, let's probably tutorials. You can just probably click on this place, tutorials. So you can see. You can read on anything. You know what Workbench. You can read. Just read a lot of tutorials, a lot of explanation. Some are PDFs, some are videos, some are videos, some are PDFs. So you can just come here, read up, read it. Sometimes I actually come here. So especially when probably, let's say probably I will say won't come here. If I when we get to this blast part, I'll come here, you can see blast. So just come and educate yourself, learn. I mean, yeah, and probably, I mean, they also organize webinars and courses sometimes. Probably they develop a new tools, or they update their tools, so you can actually hold the webinar and stuff like that. You can see. You can see called that on conferences. You can find information about it here. So, I mean, information about and see this one. So they also do. They want to have symposium. So, yeah. I think that will be the end of today's class. And tomorrow we are just going to like explore how you can search on NCBI. Please, do you have any question? Any question, please? Please, do you have any? I thought you left. All right.